So we're going to be talking today about optimizing immunotherapy and going to be used as two of our illustrative examples, both the visual vaccine, which we're looking at in a number of tumors, as well as inhibitors of the PD-1, PD-L1 axis, which clearly have now moved into first-line therapy in a number of tumors in which we're beginning to explore. So I thought I would start on this first slide with really a visual look at the endpoint that we're trying to get at. Now, this slide is actually has nothing to do with the vaccine. This is a slide which has been taken of a model using a bispecific antibody with the targeting end directed against CEA, which, as we all know, is one of the common overexpressed proteins in a number of cancers, very specifically colorectal and ovarian. And the other end of this antibody hooks to CD3 epsilon, which is part of the T-cell receptor. So it binds the cell having the targeted antigen to the T-cell and enhances the immunoeffectiveness of the T-cells. So looking at the top left slide, this is basically a control. This is not the by affinity, the bispecific antibody. This is a control, and if you look top left, you see baseline. The red area are tumor cells, and the green area are T cells. And on the top left, you see a low power view, and right below that, you see a high power view. So 24 hours after giving this control antibody, you basically see, just moving over right below where it says control TCB 24 hours, you basically still see this isolated area of tumor cells and the T cells, and there's very little overlap, and you basically get that impression when you look right down below at the magnified view, where it's really essentially no different than baseline. So let's go over now to the right and see what happens when we actually give an effective immunotherapeutic. So we start at the same baseline where you have this dissociation of the tumor cells and the cytotoxic T cells. But now, when you switch over to the right, 24 hours after using this immunotherapeutic, you basically see these tumor-infiltrating lymphocytes, what we call TILs, the cytotoxic T cells engulfing the tumor cells. And when you look down below at the magnified views from baseline over to 24 hours, you can see clearly a big difference where you now have this massive influx of infiltration of T cells. So when we go down further in the slide and you look at this little left graph, what we're looking at is the ratio over at the extreme left of T cells to tumor cells. And when you start out at baseline, you basically see, that both in the control and baseline for the immunotherapeutic, there's really no difference looking at this ratio. What percent of the cells are you looking at are T cells. But when you look at 24 hours later, you can see that just a non-specific antibody is really doing nothing, whereas the specific antibody, you're getting this huge increase of T cells directed against tumor cells. And when you look over at the extreme right, what we're actually seeing now is, as a result of this influx of these cytotoxic T cells, we're moving from baseline in the both the control and the immunized, in the group that received immunotherapy, to 24 hours, where we have this significant increase in apoptotic cell death due to the effectiveness and the influx of the cytotoxic T cells elicited by immunotherapy. So this is the endpoint of what immunotherapy is trying to do, to get a group of cytotoxic T cells, the green cells that you're seeing, in the cartoons above, to swarm into the tumor and attack the tumor cells and kill them. And this is really, I thought, an excellent visual example of what we're trying to do to tumors with our immunotherapeutic approaches. Next slide. So let's take a look at some of the ways that tumors can avoid effective immunotherapy. So first of all, we know that in order for our immunotherapy to work, we have to have something on the surface of the cancer cell for it to target. And in order to present something on the surface of the cell, it has to be processed and presented by, the HLA antigens. The mixed histocompatibility class 1 and class 2 presenting molecules. You just don't have a protein flopping on the surface of the cell. It has to be presented to the immune system by an MHC, an HLA molecule. So if that's reduced in the tumor, and a reasonable percentage of almost all tumors will have absence or aberrations of this presenting MHC class 1, that can impair immune responses. A second way is to have the tumor cells overexpressing something called Fas ligand. This is a member of the tumor necrosis factor family, and if that's overexpressed, it essentially can adhere to what's called Fas receptor on the T cell and just kill the T cells before the T cells can kill the tumor. So if you have a high Fas ligand, Fas receptor axis, you're also going to have ineffective immune control of the cancer. Another way that there can be impaired immunoresponse is what's called a suppressive paracrine effect, this number 3. And essentially what that is the tumor cells can produce these soluble products. And these soluble products can basically promote a number of inhibitory factors. It can cause the attraction of these inhibitory regulatory T cells. It can promote the attraction of these monocyte-derived suppressor cells. It can also shift the valence of some of the normal surrounding cells in the cancer environment, the macrophages and the neutrophils. There are two basic types of macrophages and neutrophils that are associated with cancer, and that's why you see TAM and TAN, tumor-associated macrophages and tumor-associated neutrophils. They're called type 1 and type 2. The type 2 essentially promote tumor growth, and the type 1 inhibit tumor growth. Well, a number of the proteins that can be secreted by the tumor cells result in type 2 inhibitory tumor-associated macrophages and tumor-associated neutrophils rather than the type 1 which we would prefer to see. Number 4, you can have dysfunctional dendritic cells or antigen-presenting cells. There are a number of secretions of tumor cells, COX-2 and prostaglandin H2, which can interfere with the maturation of these dendritic cells. The dendritic cells, essentially, the tumor cells may not be able to effectively present antigens, if they have a screwed up class 1 or, or a class 2 MHC. And what needs to happen then is the tumor cell needs an intermediary. And the proteins on the surface of the tumor cells can then be engulfed by an immature dendritic cell or an immature antigen-presenting cell. It can be then processed in this go-between. And then when the dendritic cell matures, the dendritic cell can present these proteins to the T cells. So this is another way that we may see a lack of responses to immune therapy. And then finally, and getting to the point of one of the therapeutics we're going to be discussing, there can be a deregulation of the checkpoint signaling system. Now, we talk a lot about, checkpoint inhibitors, but let's keep in mind that the immune checkpoints come in two types. There's a stimulatory checkpoint and an inhibitory checkpoint. The stimulatory checkpoints actually stimulates the production of what we call co-stimulating factors, and it's a good thing because it increases the cell's sensitivity to immune attack. However, the inhibitory immune checkpoint does the opposite. It down-regulates many of the co-stimulatory molecules or blocks them, and as a result depresses the tumor response to immunotherapeutic. So when we talk about immune checkpoint inhibitors, what we are specifically talking about are inhibitors of the inhibitory immune checkpoint. We don't really want to inhibit the stimulatory checkpoint. Let's move on. So this is something that I found in an article, which I think is a delightful way to look at our immune approach to tumor. This lists 7 factors that are important for an effective immune response to cancer. So let's start at the 12 o'clock position. You basically need tumor foreignness. In other words, the tumor has to appear as foreign to the immune system. And the reason for that is if the tumor is the same as a normal cell, if you try to do something to stimulate the immune system against the cancer, you're going to stimulate that immune system against normal cells and cause an autoimmune reaction. So you need tumor foreignness, and that is primarily as a result of cancer-specific mutations which produce proteins which are not present in normal cells. Now, going to about the 2 o'clock position, we look at the general immune status. You can't get a great immune response in somebody who's totally lymphopenic. So you need a reasonable lymphocyte count, whether that's 200 or 400. You can find articles going both ways, but you do need a reasonable number of lymphocytes and a reasonable distribution between the B lymphocytes and the T lymphocytes. Then moving down to about the 330 position, we need immune cell infiltration. You need these T cells getting in the tumor. If you think back about the first diagram I showed you, you saw a situation at baseline and with the control antibody that the tumor cells and the T cells essentially didn't overlap. There was no significant penetration. If the T cell doesn't get in the tumor, it can't kill the tumor cells. Moving down to the 5 o'clock position, you need absence of these inhibitory checkpoints, and that later on we'll get into a discussion of when we discuss PD1 and PD-L1. Over to about the 7 o'clock position, you want to get rid of those soluble inhibitors, those different cytokines that can attract these inhibitory cells. You then also at the 830 position, you have to have an absence of an inhibitory tumor metabolism. Tumors, the tumors are very dependent upon glucose utilization, and you can affect the tumor cell T lymphocyte interaction by modifying the energy level of either the tumor cell or, more importantly, the T cell. And finally, you need tumor sensitivity to the immune effector. So in other words, you have to have adequate presentation of the targeted protein on the tumor cell, which gets us to that MHC expression, what carries the protein to the surface of the cell and allows it to be presented. And you also have to have the tumor sensitive to gamma interferon, which is one of the secretions of the tumor cell, of the T cell, along with perforin and granzyme, which are basically what the T cell produces to destroy the tumor cell. So these are the components, and any one of these or any combination of these that is abnormal can interfere not only with the innate immune response against the tumor cell, but also with the therapeutic immune response. Next slide. So talking a little bit about, PD-1 blockade, the PD-1, PDL1 axis with, essentially, PD-1 in the T cells and PDL1 in the tumor cells. There's also, actually both are present in both, but we mainly talk about, PDL1, as I said, in the tumor cells and PD-1 in the T cells, that the effectiveness of using these inhibitors that we're now using in the clinic does seem to be proportional to the tumor mutational burden. Basically, we know that the oncogenes, the driving genes in many of these tumors, are basically the result of mutations, translocations, deletions, fusions, abnormalities of the genes producing these proteins. Now, when you look at all tumors and you look at the effectiveness of blocking the PD-1 or PDL1 axis, you basically can see over on the left, labeled G, that you get a very effective, durable clinical benefit, when you have a large number of what are called non-synonymous mutations per tumor. A non-synonymous mutation is a mutation that causes a difference in the protein. You can have synonymous mutations where you basically delete an amino acid, but it doesn't really affect the way the protein works or what it does. So we're looking at non-synonymous mutations which have an effect on the structure and function of the protein. And as you can see, the more non-synonymous mutations that you have per tumor, the greater the benefit of using, pembrolizumab or nivolumab, these PD-1, PDL1 blockers. And when you look over the G with all tumor and you look at the percentage of progression-free tumors, those that have a lot of these non-synonymous burdens are the most susceptible to the immunotherapy approach using blockade of the PD-1 axis, as opposed to those that have a low number of the total mutational burden. Next slide. Now, we've seen this slide before, and it's not the fact that the total mutational burden, the number of mutations, that's that important. What's important is the fact that the more non-synonymous mutations we have, the more likely we are going to have what we call neoantigens. A neoantigen is basically a small peptide string, about 7 to 9 amino acids in length, that is an epitope. In other words, it's capable of being presented on the surface of the tumor cells, highly bound to an MHC, and then once that neoantigen is bound to that MHC, it will have a strong binding to a T cell receptor to cause a linkage, and then that T cell can begin to attack the tumor cell. So the more somatic mutations we have, the more of these non-synonymous mutations we have, the greater the likelihood we're going to have neoantigens. So when you look all the way over on the right, you'll see that the greatest number of neoantigens are on melanomas, which are related to what? Ultraviolet light and a lot of mutations. Lung cancer, which is related to smoking, and thus a lot of mutations. Colorectal cancer, which certainly in large part is environmentally caused based upon some of the things we eat, and has a lot of mutations. So these are more likely to have a large number of neoantigens. Over on the left side, you have a number of leukemias and lymphomas. You also have, which isn't completely included on this slide, a lot of the pediatric cancers. Ewing Sarcoma has one of the lowest tumor mutation burdens in all of cancers. Now, does that mean it doesn't have a neoantigen? No, but it just means it doesn't have a whole lot of neoantigens. So let's move on to the next slide. Now, we keep talking about antigens, but I just want to remind everybody there are different types of antigens, and this slide will express the importance of the neoantigen. So over on the top, under category on the left, it says overexpressed gene products. So we know that in cancers, a lot of the genes are amplified, they're amplified. You have, more than these normal number of genes, and produces a lot more of these surface proteins. The problem with this is, even though that there's a lot more of these proteins on the surface of the cancer cells, these are just overexpressed, which means they're normally expressed on regular cells. And as a result of that, there's a reasonable chance that you're going to be having the possibility and a high likelihood of resulting in an autoimmune reaction. You can then have a group of antigens or cell surface proteins, which are called tissue-specific differentiated antigens. An example of this, for instance, would be PSMA. A prostate-specific membrane antigen in prostate cancer, which essentially is present in prostate cancer, but mainly in normal prostate. Now, you can use these. The problem with these is that even though they're mainly present only in very limited normal tissue, they can also still be present in lower amounts in normal tissue. So there's still a possibility of eliciting an autoimmune reaction. You then can have cancer germline antigens. These are proteins that are presented on cancers because cancers de-differentiate. They go back to a more primitive state, and they re-express proteins that are normally only present on fetal cells. And these can be high in cancer cells, and generally they are not expressed in adult tissues. The reason they're not expressed is because they're normally expressed in testes, and there's no MHC presentation group in the testes, so that these antigens are normally not presented during fetal growth, so that the body doesn't become tolerized to that. Now, the problem with the cancer germline antigens, even though that they can elicit a relatively specific immune response, is some of them actually are important in certain areas of the adult. For instance, if you look under cancer germline antigens where it says the MAGE-8 family, well, one of the MAGE-8 family is called MAGE-3. And MAGE-3 happens to be very important in our brains. And if you start immunizing against this germline antigen, it can cause significant autoimmune responses against some of the cells within the brain. So one even has to be very careful with these. And then we basically get to some of the antigens that are now truly becoming cancer-specific. One are viral antigens, and these are normally not expressed in normal tissue. They essentially aren't expressed in normal tissue, but they can be expressed in certain tumors that have a viral etiology, for instance, cervical cancer and squamous cell carcinoma of the anus, which are related to human papillomavirus E6 and E7. And we know that young girls are now being immunized against some of the PV serotypes in order to try to prevent cervical cancer. And there actually is some data that this can also be preventative for some anal cancers, as well as some of the P16 positive head and neck cancers of the tonsil and the base of tongue. But finally, we get to these cancer-specific mutated antigens, the neoantigens. And these are just not present at all in any normal cell. And those are KRAS mutations in colorectal and pancreatic cancers, the EWS-FLE1 fusion gene in Ewing sarcoma. These just aren't expressed in normal tissue. So if you can immunize against these, you are specifically targeting cancer cells with almost zero risk of causing an autoimmune reaction. Now, when we talk about these mutated antigens, we should divide them in two types. There are a number of mutated antigens that are not driver genes. They're mutated in the cancer, they're not present in normal tissue, but they're not necessary for the cancer to continue growing. Then there are mutated antigens which are still specific to the cancer, but are absolutely necessary for that cancer to continue to thrive. Those are called antigens from driver genes. Now, what you want to do is you want to immunize the patient against the neoantigen from the driver genes, because the tumor can't just easily get rid of these proteins. If you immunize against a non-driver gene, the tumor can basically defend itself by saying, well, this isn't really necessary, so I'll basically cover it up, I won't express it. But it can't do that with a protein from a driver gene. So as a result, that's basically what we want to be able to target. Next. This is a busy slide, but the only thing, just look at the bottom. Later on, you could look at this at your leisure. And all this shows, based on what we were talking, is that tumors that have DNA repair defects, where there's a higher probability of having large numbers of tumor-specific mutations, tend to have the best response to anti-PD-L1 and anti-PD-1s. And if you, later on, when you look through the specifics of the different organs, you can basically see that there is a nice relationship between DNA repair defects and the effectiveness of pembrolizumab and, nivolumab. Next slide. Now, how do we begin to identify these things? This is something that we're going to be doing in the clinic. So let me walk you through this, starting over at the left, where we basically have, you start out with the somatic mutation, and that we know we can identify. We've been sending stuff off to foundations for whole gene sequencing and whole exome sequencing. So that basically we're already doing to identify antigens. And what you want to do is you want to identify these mutations in the tumor cells. You want to say, okay, I'm identifying these mutations. I'm also looking at normal cells, and I'm picking out these mutations in the cancer cells that are not present in the normal cells. So that we do with, as I said, whole exome and home genome sequencing. And then we know that those have to be, transcribed into messenger RNA. Now, it's important to look at the messenger RNA. Why? Because you can have a cancer-specific mutation, but if it's not expressed, if it doesn't produce a target protein, it's worthless in terms of trying to immunize against it. So one screen is you start out knowing the mutations, but then you're looking to see, by looking at the mRNA, is it being expressed? And that we can easily do with RNA sequencing. Then once you have that, we know that the next step is to get the proteins produced by the mRNA. It gets translated into proteins. That essentially has to go through the proteasome, and that protein has to be broken down into little strands of peptides. And it's going to be one of these peptide strands, these components, 7 to 9 long strands, that are eventually going to get to the surface of the cell. Now, there are algorithms to figure out which are the most likely groups of these 7 to 9 component strands are produced. And it's important because there are two types of proteasome in the cell. There's a standard and an immune proteasome. And we're really interested in what the immune proteasome can produce. But the algorithms are now relatively effective in identifying these. Then you have to get these 7 to 9 long peptides. You have to basically get them into the endoplasmic reticulum and then to the surface. And there's something called the TAP system, which is the transporters associated with antigen processing, and there are algorithms to figure out which of these peptides are most amenable to TAP transport. Then we get to number 5, which is the binding of these things to that MHC class 1 presentation molecule. And there are more algorithms available to be able to determine which of these peptides has the greatest affinity, the greatest magnetism for the HLA, the MHC, presenting molecules. And then finally, we get to the T-cell receptor. And what we're essentially looking for is another algorithm and some other evolving ways in which to measure the affinity, the binding affinity between the MHC holding on to the peptide connecting to the T-cell receptor that's going to activate the T-cell, turn it on, and allow it to become activated so as to attack the tumor cell. So this is what we have to go through to find out what exactly is being sensitized and how, by identifying the exact neoantigen that is triggering the immune response, by isolating it, we can then upregulate it, increase the cells that are able to recognize it, and make our immune approach more effective. Next. Now, one of the things that's interesting is when you look at immunization, the tumor cell just doesn't sit idly by. When you try to immunize against the cancer and you start getting a bit of a T-cell reaction, if you look at the top left, what you basically see, you see this red, green, and kind of orangey or sienna-colored squiggle. Well, what you're basically seeing is as you get more of a T-cell response against the tumor, what tends to happen is the tumor adapts. You have what's called an adaptive resistance. So what you're seeing at the top left is you're seeing a shift that with time, as a result of having activated T cells against the tumor, the T cells secrete interferon gamma. The interferon gamma stimulates the tumor cell, and you wind up having a much higher level of PD-1, and the higher the level of the PD-1, the less likely it's going to be sensitive now to the incoming T cell. So even though you're stimulating an immune response against the cells begin to have a way of defending themselves by countering the response of the T cell. Now, if you look at all these different things, you'll see that it's really PD-1 that, in terms of what is most likely to mediate the inhibitory response, is the major player. CTLA-4 and LAG-3, again, they're slightly increased, but nothing that's showing an evolution over time. Some of the ones that do show an evolution are showing no difference between baseline and 3 hours and 24 hours. So even though there's some non-specific effect, there's not an antigen-specific effect for most of these other than PD-1, which gives you the importance of blocking the PD-1, PD-L1 axis. Next. So what adaptive resistance? Let me just repeat it. You have your CD8. That's a T cell, a cytotoxic T cell. It's been sensitized against the cancer. It produces gamma interferon. It comes into the attack. Then you switch over to B, and because of the attack and the release of gamma interferon, it stimulates the cancer cell to make PD-L1. And as a result of PD-L1, you wind up having kind of a shield around the cancer cell because you now have this block, this PD-1, T cell PD-1, tumor cell PD-L1, and because you've elicited this immune checkpoint inhibitor, you are now inhibiting the immune response. So what initially might have had some effect has an equally strong and overpowering counter effect. And that's what we are looking at to block and reverse by using drugs like nivolumab and pembrolizumab. Next. Next. Now, one of the interesting things when we go about identifying all of these is we can get our T cells from two sources. We can get them from the T cells in the cancer, the tumor infiltrating lymphocytes, or the circulating T cells, the peripheral blood mononuclear cells, but we have to be very careful. So if you look at this slide, which comes from 24 melanoma patients, that Gross and colleagues published, just about two years ago. Look over at PD-1 expression, and look at the fact that if you're measuring PD-1 expression in lymphocytes and you're using the tumor-infiltrating lymphocytes, you're up at about 30%. But if you're measuring peripheral blood mononuclear cells, even though they may have the same responsiveness to antigens, there's a much lower probability of them demonstrating PD-1. And if you look across the board, you'll see everything where there are 3 stars shows a difference between cells. And when you look at the PD-1, the LAG3, the TIM3, all of these basically are inhibitory immune checkpoints, and if you notice, they all tend to be higher in TIL cells than in peripheral blood mononuclear cells. So ideally, what you want to measure are the tumor-infiltrating lymphocytes to get the best feel for what is characterizing the immune-activated T cells. Now, over on the right, we begin to look at what we call memory cells. There are different types of memory cells. The reason we're interested in memory cells is, one, these are cells that are measured by the ELISpot, which we use in our Vigil vaccine. And most of the memory cells that are important to us are what are called the effector memory cells. So when you look over to the right, you're going to see a whole bunch of numbers and designations. But the first group that you see, the CD62L positive and the RO negative, those are naive. Those haven't been stimulated yet. When you look over at the 62L positive, RO positive, you're looking at what are called central memory cells. Those are cells that do serve a purpose. They eventually will have something to do with secondary responses, but there's not that much difference between TILs and peripheral blood mononuclear cells. But when you look over at the 62L negative, RO positive, those are the effective memory cells. Those are the ones that are mainly measured by the ELISpot. And those are the ones that you'll see are much higher in TILs than peripheral blood. Now, we all know when we look at our patients, we're looking at peripheral blood. But if we were measuring TILs in the tumors, we might find that there are a much higher level of these effective memory cells. So when we look at the changes in ELISpot test in peripheral blood, it may not be the best reflection of what's actually going on at the site of the tumor. If you look all the way over to the right, that just goes to show you that most of the lymphocytes, whether they're TILs or peripheral blood mononuclear cells, present one of the inhibitory checkpoint inhibitors, whether it's PD-1, TIM3, or LAG3. Some of them, however, will present 2 of these together. Some of them will present 3 of them together. The reason this is important is that knowing there are other checkpoint inhibitors, there are other things that can be done if blocking PD-1 isn't effective. PD-1 may not be effective because you have a high TIM3 or a high LAD3. 4-IBB, just to throw out at you, is a stimulatory checkpoint. So if that's high, it's great. Next. Next. So, we talk about, using, PD-1 expression in order to, determine how likely it is that a tumor is going to respond to one of the checkpoint inhibitors, pembrolizumab or nivolumab. And indeed, we know if it's PD-1 positive, as you can see on the left, if you're measuring gamma interferon, which is what we measure in the Ellis spot, those are pretty high. Now, CD137, I told you, is essentially a stimulatory immune checkpoint. It's the same thing as 4-1BB. Now, if you look over at the right at CD137 negative PD-1 positive, you'll see that even though you'll have a group of cells with a PD-1 positive, if they're CD130 negative, they're not really going to be producing a whole lot of interferon gamma. However, you can have cells that are PD-1 negative, but if they express CD137, which is a positive immune checkpoint, you actually can have high interferon gamma. So we do have to be careful as we start figuring out what tumors are likely or less likely to respond to some of our therapeutics by realizing that there's more in life than measuring PD-1 and PD-L1. Next. Next. So let's get very specific right now. Over on the left is that septogram I showed you before, where we had all the parameters that allow for effective immunotherapy. Over on the top right, we're seeing those, 7, necessary components for immune response, with 1 being a problem. And that basically is the absence of checkpoints, PD-L2 in this case. So what do we want to do? We want to correct that. And by correcting that, we can get a much more effective immune response. And looking at this immunogram, we can, for each tumor, theoretically, we can figure out what way is the best way to approach this tumor in terms of the immune therapy that we're going to be using. In this particular case, we're looking at the the PD-1, PD-L1, L2 axis. So next slide. So looking at PD-1, PD-L1, next slide. We can subdivide our tumors into 4 groups. And remember, this is only on the basis of PD-L1 expression, something that a lot of people are now doing to decide, how helpful will it be to use, pembrolizumab, for example, in a particular tumor. So type 1, over in the top left, has a lot of TIL, and you also have a high expression of PD-L1. And this is when you have adaptive immune resistance. In other words, you have a lot of PD-L1 because you had a lot of infiltrating T-cell lymphocytes with the counteracting expression of PD-L1. Over on the top right, you have a situation where you have no significant tumor-infiltrating lymphocytes, and you don't have PD-L1. Well, that's immunologic ignorance. That means that these tumor cells aren't recognized by the immune system at all. You go down to the bottom right, and you have these type 3 cases where you have PD-L1 made by the tumor, but you don't have any infiltrating T cells. The reason for this is, in addition to the tumor cell producing PD-L1 as a response to these incoming T cells, there are certain gene mutations in the cancer which can result in the production of PD-L1, whether or not you have TIL cells. So that's type 3. And type 4 are when you have TILs, but essentially you don't have any PD-L1. And that basically is due to what we call tolerance, or more likely when you have other suppressors like LAG3 or TIM3 present. So taking each of these different components, let's take a quick look at how we can approach them based upon our understanding of the immune mechanism. So the next slide. So let's look at the type 1, where you now have this adaptive immune resistance. This is the one that basically has a lot of TILs and the tumor cells express PD-L1. Well, this is a tumor likely to benefit from, blocking that using pembrolizumab. Now, you may want to look quantitatively, how much, what's the percentage of TILs, how much PD-L1. We know that we have antibodies against PD-1 and PD-L1, and they do work slightly differently. You may actually find in certain situations it's worthwhile combining them. I won't go into the specifics, but they do work slightly differently. You also then have the possibility of vaccine enhancing this process, because when you treat with a vaccine like Vigil, and we have good data on the GM-CSF producing autologous tumor cells previously used called GVAX, that there are both murine models and clinical demonstrations that following immunization, you actually can elicit an increase in tumor-infiltrating lymphocytes, as well as an increase in PD-L1 expression of the tumor cell. So vaccination can certainly enhance the effectiveness of pembrolizumab and other of these inhibitor checkpoint inhibitors. And then by combining judiciously with radiotherapy, chemotherapy, and targeted therapy, a lot of these combinations will also result in an increase in PD-L1 or an increase in tumor death that will expose new antigens and attract more T cells. So there are multiple options we have for this type 1 situation. Next slide. For the type 2 situation, this is where we have immune ignorance, where we just don't, the body's not recognizing anything having to do with the tumor. So this is a situation where there's data that's now suggesting that maybe if you combine blocking, PD-1 and blocking CTLA-4, pembrolizumab, that combination may be effective in a certain proportion of patients with this particular situation, this type 2 situation. We know that using certain types of immune stimulants, poly-IC or STING agonists, to stimulate alternative routes of immune expression can be effective. We are hoping and looking in one of our protocols to see whether Vigil in this situation can effectively increase TILs, and as a result of increasing these tumor-infiltrating lymphocytes, increase PD-L1. This is a good situation where the CAR-specific T cells, adoptive immunotherapy, where you take activated T cells, target against a certain presenting antigen, even though the patient didn't recognize it. These T cells taken from outside sources and specifically targeted against the tumor cells might be able to recognize it. And this is also a situation where when you might be able to use radiation chemotherapy and or targeted therapy, which by having effects on the tumor cells may be able to make them more immunogenic in the sense of being recognized by the immune system. Let's move on to the type 3 situation. And the type 3 situation was intrinsic induction. That's where there's essentially no TIL, but because of one of the mutations in the tumor cells just producing PD-L1. So, you know, what you want to do in this situation is you want to get TILs in there, and you can do that by the same poly-IC, the inosine cytosine polymers or sting agonists in order to try to get TILs in. We're looking again at Vigil to see if we can get the TILs in. One way of getting TILs in is adoptive therapy using these, chimeric antigen receptor specific T cells. So even in this group, there are ways that we can use or have the potential of eliciting an immune response against these tumors. And once again, combined therapy with our standard core of therapeutics might be able to elicit some type of immune response by damaging these cells and having them release certain products which are more effective in pulling in TILs. Next. And the final are basically immune tolerance, where you have TILs but no PD-L1. I had mentioned that there may be other checkpoint inhibitors to look at. We may want to target LAG3. We may want to target TIM3. IDO can be overexpressed in these. All of these are basically proteins that will inhibit an immune response. We can try to, I mentioned earlier on that there are two types of macrophage and two types of neutrophils, and we want to increase the type 1 TAM and the type 1 TAN over the type 2, because the type 1s are essentially the ones that will attack the tumor cells. And there are ways that can be looked at and that are being looked at. Zoledronic acid can impair TAM maturation and enhance T1. Trabectin, which is a chemotherapy agent, can inhibit a chemokine which attracts type 2s. GM-CSF, which is a component of our Vigil, induces the tumor-associated macrophage 1 polarization, and blocking TGF-beta, which Vigil does, actually increases the percentage of surrounding TAM1s over TAM2s. So clearly, given that Vigil both secretes GM-CSF and blocks TGF-beta, it's kind of a no-brainer that we would want to look at it in this type. And once again, we can fall back on, a kind of off-target effect of radiotherapy, chemotherapy, and targeted therapy to try to make these cells much more immunogenic. So that basically is kind of an overall introduction to, how we can, work with and modify the immune system, with the end of what we've just discussed, really zeroing in a little bit more on both our Vigil vaccine and what's available to us now in the clinic with the PD-1, PD-L1, and even CTLA-4 inhibitors. So I'll stop there.
Neil, I thought that was really cool. I mean, that was a really, really good summary. It sort of highlights, just as we're trying to identify the upstream molecular signal pathways and in essence categorize patients into potentially sensitive and potentially resistant groups based on the molecular profiling. Your breakdown of the 4 types of immune situations that cancer cells may be in, I'm wondering if we can identify the best way that we can characterize patients into one of those 4 categories. We've got the PD-1, PD-L1 expression, but it sounds like we want to try to fit patients, or at least characterize them according to one of those 4 types, wouldn't you think?
Mm-hmm. Yeah, and it's, once you get the tumor, essentially the categorization, at least in terms of, this algorithm is looking at TIL infiltration and PD-L1 expression. Now, the relative amounts, the frequencies, that becomes a different question. But, those are just looking at two parameters. And with those tumor parameters, you basically have at least a general way of getting 4 subtypes, which we probably need to think of in terms of somewhat different immunotherapeutic approaches.
Yeah, so what we can potentially do is hook up with one of our path labs, ProPath, potentially, and let's see what kind of program we can work out with them to potentially characterize. Ideally, it would be with a a core biopsy opportunity of a patient coming into us. When they start to come in and they need, you don't have a lot of chance for attempting multiple therapeutic attacks. We need to hit this patient early when they come to us. It may just be that we need to do a biopsy on patients coming in. We've started off getting the molecular profile. A lot of that's from archival tissue, but if we really want to be efficient, we should get a fresh biopsy, characterize TIL, characterize PD-L1, PD-1, but that would come into the molecular signaling. I'm wondering if we should start to consider a fresh biopsy literally in every patient comes into our clinic in order to sort them out according to these categories and get the fresh molecular database. You know what I mean?
Yeah, and we can begin to do that, but we also have to keep in mind that as you divide, these into 4, different patterns based upon these 2 parameters, that the therapeutic approaches I listed, are therapeutic approaches that make sense, but most of them, haven't really been tried. Theoretically, they make sense. Some of them have been tried. But, we have to realize that we're still talking here about likelihood and probabilities and theory as opposed to a whole lot of experience.
Yeah, and we are where you would develop that experience. Other sites won't be doing this. And, if you're that patient and you have a technology that is developed based on a mechanism that you have demonstrated in order to get that product to the clinic, it seems to make sense to at least put that agent with that mechanism into the environment that says that mechanism is effective. You know what I mean?
Right.
Just like you're saying. So, it's not something, and we've gone through this with the molecular side, and we've now seen, we're now already seeing fruits of it. This is something I think, Neil, you and I can start to work out some programs, but I can check out, various labs. Megan maybe will help me with this as well on how efficient they are in the TIL assessment. I think the PD-L1 is a piece of cake for us. That'll just be the molecular profile. But the TIL is what I'd like to have a good handle on. So maybe we should sort that out. That's a great.
But we can't forget the fact that these last 4 slides were essentially modeled on a situation where the major problem in terms of an effective immune response are the checkpoint expression. But we have to keep in mind that some tumors won't have MHC expression, and that becomes a different problem. Some tumors, some patients are just totally lymphopenic because of prior therapy, and there's no way that it doesn't much matter what we do on them in terms of some of the things we're talking about. Some of the patients, they have a ineffective interferon gamma expression, and that can be a problem that may not be addressed by any of these. So we can't lose sight of the fact that even though we may be able to help a reasonable percentage of patients, that there are defects in the the immunogram representing the patient's totality of immunologic capabilities that need to be kept in the back of our minds, that there are other factors despite PD-L1 and TIL that may still be problematic.
Oh, absolutely. And part of that
Right.
is covered, as we've talked about with the JAK mutation. We'll get it on the molecular
Right.
profile. And then our recourse is always, we don't have to force our patients onto an immunotherapy. We've still got the targeted opportunities. It's going to be a balance of
Right.
what targeted signals they have and what immune. And we still have a lot unknown, but this is a lot better to me to go by this guideline or to try to develop a program along these lines. It took us 2 years to get the molecular thing kicked in, and we're still, not optimal, but we're so far ahead of other clinics that it doesn't seem to be bad to sort this out this year.
No, it doesn't. Not at all.
Yeah. Cool. Are there other questions from anyone else? Well, cool. Well, thanks, Neil. I appreciate it. These slides are great, too, by the way. Thanks a lot.
You are welcome.
I'll see you later, man.
Great day, everybody.
Dr. Senzer.
Bye.
Bye.
Yeah. Bye.
Bye.